Hi, we'll be exploring the two powerful methods, short-time Fourier transform, which is known as STFT, and wavelet transform, which is known as the WT. So these crucial techniques are crucial for understanding the muscle, activity, and fatigue. STFT breaks down EMG signal into short segments, while the WT provides a multi-resolution analysis, particularly useful for a non-stationary signal.
As for the data acquisition, we use Nina Pro. So basically, Nina Pro is an online website where you can use to find any biological signal, especially EMG. So the reason why we use Nina Pro is because it is easy to obtain signals and quite straightforward. The data also represent in MATLAB, which is quite easy to use on MATLAB. And here I also include the link for the website. An explanation for signal that we have obtained is we chose subject number 15, which is a female, 26 years old, having right hand as her dominant hand. So for the device used in this experiments is a 10 Autobot Myobok electrodes, which is used to identify the SEMG signals or the surface electromyogram dataset. And each of the subject perform three kind of exercise. Number one is basic movement of the fingers, and then we have isometric hand movement and also functional movement. In this case, for the assignment, we chose subject number 15 to perform exercise number 3, which are grasping and functional movements. So the procedure for this experiment is that subjects are asked to mimic the movement shown on the laptop. So the movement that they have done is basically already been listed on this list. So basically, movement number 1 until number 23 is only specifically for exercise number 3, and each of these movement of the obtained signal will be recorded on the 10 electrodes of the myobok. So for this assignment, we specifically analyze the electrode number 8 or channel number 8 of the myobok. The location of the electrode number 8 is around the forearm of the subject or highlighted in red, as you can see from the diagram. So this is the SEMG signal obtained from the 8 electrode of the myobok from subject number 15 performing exercise number 3, which are grasping and also functional movements. To be noted here that the signal has a sampling frequency of 1000 hertz as a default. There are literature review here explaining the purpose of the EMG recordings on the Nina Pro website. The one being to support research on advanced myoelectric hand prosthetics, where the database is obtained from the SEMG of the forearm and also the kinematics of the hand and wrist of the subjects while performing predefined actions. The second being is that to allow research groups to develop and having test movement recognition and setting the benchmark for scientific database, especially in SCMG or biological signals. The third being building the Nina Pro database, which is a resource for biorobotics community.
Okay, now I'm going to explain about the short-time Fourier transform. The purpose of the STFT is to use for analyzing how the frequency content of a signal evolves over time by breaking the signal into shorts. Then the Fourier transform is applied to each segment to produce a time frequency representation. So the following is showing the parameters used, which is the sampling frequency, the window type, window size, overlap percentage, as well as the FFT length. So this is the steps for analysis the STFT. So after that, the coding is shows decay variables EMG channel FS, window, overlap, FFT lamps, and the spectrogram. And then the output log EMG data from file will generated, and the input EMG data was stored in variable. Then select the channel A and confirm the sampling frequency, which is 1000 hertz, and then set the STFT parameters. Then the output spectrogram will successfully generated. So the spectrogram result shows that the frequency range is between 0 to 50 Hz, which suggesting the slow muscle contractions or steady motor unit activities, while the temporal trend shows notable bursts of high power at certain time intervals, which indicate the muscle activity. The dominant frequency shows higher power in lower frequency bands corresponds to slower muscle contraction.
So for my part, I'll be explaining the steps involved in wavelet analysis. So to analyze the EMG signal using wavelet methods, the following steps were performed. First, one channel of EMG data was selected and visualized in the time domain to understand the raw signal. A sampling frequency was selected to ensure accuracy. Next, the wavelet decomposition was carried out using the DP4 wavelet analysis, which is widely used for biomedical signals due to its ability to capture subtle signal details. The signal was decomposed into 5 levels, enabling the study of frequency component at multiple resolution. So after decomposition, the detail coefficient for each level were extracted. These coefficients represent different frequency bands, helping to isolate patterns or noise. The detail coefficients for all levels were then plotted to observe the step-by-step signal breakdown. Finally, the results were analyzed by comparing the original signal with its decomposed components. This decomposition provides a clear breakdown of the signal, allowing for the study of specific features such as bursts of activity, low frequency trends, and effective noise filtering. So for this slide, this is the original EMG signal captured from a single channel. The x-axis represents time in seconds and the y-axis shows the amplitude. The data was sampled at 1000 Hz. You can see a mix of high frequency components which could indicate muscle activity and some potential noise. So here we have the results of the wavelet decomposition performed on the EMG signal. We use DB4 to break the signal into 5 levels of details. The first plot at the top is the original signal. The subsequent plots show the detail coefficients as for level 1 through 5, representing finer to coarser details of the signal. So we observe as we move the higher levels, we see a more smoothed out version of the signal, highlighting low frequency components. This layered breakdown allows us to identify distinct signal characteristic at different scales. So finally, the decomposition provides several insights. At level 1, the detail coefficient reveal high frequency bursts that could indicate muscle activity or noise. At higher levels, such as level 3 through 5, we see smoother components that likely correspond to the overall trends of muscle activation. This ability to separate noise from meaningful signals features make wavelet analysis a powerful tool for analyzing EMG data.
As for the discussion, the observation from short time Fourier transform, the spectrogram here shows two colors, which are yellow for high amplitude and blue for low amplitudes. The spectrogram shows clear concentration of yellow on lower frequency, which means high energy at low frequency, which is typical for EMG signals. Lower resolution in high frequency make it less effective for isolating noise and also short muscle bursts. SDFT is more preferred to understand the whole behavior of the raw data over time. As for the wavelet transform, the details level highlight noise and progressively smoother trends at lower frequency. The advantage of WT is that it able to denoise the signal by reconstructing the raw data and removing the high frequency components, which is in this case detail level 1. And then WT is more preferred to analyze localized bursts, remove noise, or extract features. As for the comparison between STFT and WT, time frequency resolution for STFT is fixed while WT is adaptive. That's why we saw wavelet transform has a lot of waveform rather than spectrogram only one. And then the signal application for STFT better for stationary signals, while wavelet is much ideal for non-stationary signals. As for the noise handling, short time Fourier transform has a problem where the noise is mixed into the spectrogram, while wavelet able to handle it by isolating it in specific levels. And the computations, STFT has much more simpler and faster method than wavelet, where it is much more complex. So that is the comparison between these two techniques.
Alright, so our key findings reveal significant muscle activity in the 0 to 50 Hz range using STFT with temporal changes detected through power bursts. Wavelet analysis showed high frequency component at level 1 and underlying low frequency patterns at level 3 to 5. We also use specific processing parameters, including a 1000 Hz sampling frequency and a Hamming window with 50% overlap to ensure accurate results. The combination of STFT and WT provides a comprehensive view of EMG signals, offering both time frequency representation and multi resolutional analysis. This dual approach would yield deeper insights into muscle activation patterns with potential application in biomechanics, rehabilitation, and sports science. In a nutshell, these advanced signal processing techniques actually prove valuable for analyzing complex biological signals like EMG, opening new avenues for research and clinical application. All right, thank you.